Chromatography. Chromatography obtained from two Greek words, chroma and graph. Chroma means color, graph means writing. Chromatography is a valuable technique for the separation and purification of small samples of mixtures. As the name suggests, this technique was originally confined to the separation of colored substances, such as plant pigments or dyestuffs. The name is no longer accurate, as the technique is now equally well applied to colorless substances. Chromatography is based on the principle of selective distribution of the components of a mixture between two phases, a stationary phase and a moving phase. The stationary phase can be a solid or liquid, while the moving phase is a liquid or a gas. When the stationary phase is solid, the basis is adsorption, and when it is a liquid, the basis is partition. Some of the common types of chromatography are column chromatography, thin layer chromatography, paper chromatography, gas chromatography. Column chromatography. In column chromatography, the powdered adsorbent, may be alumina or silica gel, is packed into a vertical glass tube. A small quantity of the mixture to be separated is added to the top of the column, where it remains adsorbed. A solvent, pentane, benzene, ether, is then allowed to flow down the tube. Separation of the components of the mixture takes place as a result of differential adsorption. If the components are visible, colored bands appear down the column. The flow of the solvent is continued until all the components are eluted from the bottom one by one. The different fractions are collected and individual components recovered from the solution. Thin layer chromatography. Thin layer chromatography, it is possible to separate even minute quantities of mixtures. A sheet of glass is coated with a thin layer of adsorbent, maybe cellulose powder, alumina, silica. This is done by mixing the adsorbent with a suitable liquid, usually water, to form a slurry. This is applied to the glass by spreading or dipping. After drying the plate, a drop of the mixture is placed just above one edge, which is then dipped in a pool of solvent. The solvent is drawn up the adsorbent layer by capillary action. The separation of the mixture occurs as in column chromatography. Paper chromatography. The process is similar to thin layer chromatography, except that the strip of paper acts as the adsorbent. Gas chromatography. Gas chromatography is used to separate and analyze mixtures of volatile organic liquids. The column packing consists of porous, inert powder impregnated with an oil, maybe silicone or petroleum jelly. Helium is passed through the heated column, and a sample of the liquid mixture to be separated is injected into the helium stream. The mixture vaporizes and is carried through the column by the helium flow. The components of the mixture pass through the column at different rates, some quickly, some slowly. As each component reaches the detector, A peak is provided by the recorder. The area under the peak is proportional to the concentration of that component in the sample. A component is identified by its retention time, that is, the length of time it remains in the column. By chilling the exit stream of helium at appropriate times, each individual component can be condensed and collected. Tests of purity. A pure organic substance has characteristic physical properties: crystalline form, refractive index, specific gravity, melting point and boiling point. If a given sample of substance shows the properties that the pure compound is known as to possess, it may be considered pure. The numerical value of these properties changes with the nature of the amount of impurities present. In most laboratory work, the melting point of a solid substance and the boiling point of a liquid substance is considered a sufficient indication of its purity. Melting point. A pure solid substance melts sharply at a definite temperature, while an impure substance will have a lower and indefinite melting point. Mixed melting point. The method of mixed melting points as a means of establishing the purity of a substance depends upon the fact that a pure organic compound possesses a sharp melting point, while the presence of impurities usually lowers the melting point. Boiling point. A pure organic liquid boils at a fixed temperature, which is characteristic of that substance.